This is our cell cycle. We start with interphase, which is the longest step in the cell cycle, consisting of 3 different phases in which the cell acquires nutrients, creates and uses proteins, grows, and replicates DNA. The first stage is the G1 phase, in which the cells increase in size, provides as a growth phase for RNA, and each of the chromosomes are with a single DNA molecule. Before the cell can pass to the S phase, it must complete the G1 checkpoint, where the cell is checked for appropriate nutrients, correct growth factors in size, DNA damage, and proteins. If it does not pass this, it goes to G0, where the cell will rest until it's ready. If it does pass, it will go to the S phase, where DNA replication occurs, as well as DNA synthesis. The amount of DNA will be doubled and chromosomes turned to sister chromatids. It will then go to the S checkpoint, where it is ensured DNA is replicated correctly. It will continue the G2 phase, which is the gap between synthesis and division. It grows as it prepares to divide. At the G2 checkpoint, the cell is checked for DNA damage and completeness. If all of this is completed, the cell will move on to mitosis or M phase. During prophase, chromosomes condense and become visible. Centromeres made up of 2 centrioles move towards the opposite poles to form spindle fibers. Spindle fibers emerge from the centrosomes, and the nuclear envelope breaks down. Spindle fibers form a protein structure that divides genetic material in a cell. Metaphase. During metaphase, chromosomes line up at the metaphase plate. Each sister chromatid is attached to a spindle fiber originating from opposite poles. By now, the nucleus has been dissolved, and chromosomes have condensed together to line up in the middle. Anaphase. During anaphase, centromeres split into 2, and sister chromatids are pulled towards opposite poles, while spindle fibers elongate the cell. Each chromatid becomes an individual chromosome.
During telophase, chromosomes at opposite poles decondense and nuclear envelope material surrounds the chromosomes. During cytokinesis, animal cells use cleavage furrows to separate daughter cells, and plant cells use cell plate to separate them. This final event makes sure that the nuclear and cytoplasmic contents are right between the cells.